This video will be regarding the biology and key features of Alzheimer's disease and how clinical research and development is required for new treatments to become available through the clinical trial process. It is advantageous to learn more about the disease that is affecting one person out of every 79 in the UK and how to be more informed about the clinical research progress in this area. Alzheimer's disease. What is the difference between Alzheimer's disease and dementia? Dementia is not a disease itself, but rather a set of symptoms that result from damage to the brain. Symptoms include memory loss, personality changes, and impaired reasoning. There are a few common forms of dementia, including Alzheimer's disease. Alzheimer's disease is the most common type of dementia and is caused by tangles and plaques forming in the brain. Progression occurs over years, and there are two main types, late onset and early onset. How does the brain change in Alzheimer's disease? The brain is comprised of specific cells called neurons. These have roles in the nervous system to transfer messages to other neurons or cells to cause an action, for example, a muscle to contract. The physiological role becomes damaged in Alzheimer's disease due to two different hallmarks. The first hallmark is neurofibril tangles, NFTs. These are located predominantly in the axons of neurons. Microtubules are structures that are inside cells that can extend or shorten to move nutrients, cell components, or molecules throughout a cell. Microtubules use a protein called tau to assist with its stability. When the protein is phosphorylated in Alzheimer's disease, the change causes tau not to bind to the microtubule, and instead it binds to other tau proteins. This aggregation forms NFTs. As the microtubules are no longer stable, they break apart and no longer work, reducing the function of the neuron. The second hallmark is amyloid plaques. Cells create a protein called amyloid precursor protein, APP. This is normally broken down into proteins that assist with neuronal growth and repair. In AD, a genetic mutation changes how APP is broken down and produces a mutated sticky form of the amyloid beta protein. The mutated form of amyloid beta is small enough to leave the neuron. Its sticky structure binds to other amyloid betas and aggregates. This aggregation begins small but will soon form a plaque. Plaques can be forming into the hippocampus even up to 10 years before any signs or symptoms. The amyloid beta is also very toxic to neurons, and the buildup can cause neuronal death. Risk factors. What does this mean? A risk factor is a variable that is associated with the increased risk of disease or infection. Please note the relative percentage of increased risk can be very small. There are two main types of risk factors, modifiable and non-modifiable. For AD, there are several non-modifiable risk factors. The first is age, with an increased risk for over 65s, ethnicity. South Asian, African, or African Caribbean origins are more susceptible. Genetics. Several genes have been identified with a link to AD, including the apolipoprotein E and the APP protein. Gender. Women are more susceptible than men. And finally, head trauma. Damage to neurons and becoming oxygen deprived could speed up degeneration. There are three main, potentially modifiable risk factors for AD. The first is cardiovascular disease, which is impacted by lifestyle choices such as smoking, inactivity, and high cholesterol. The next is physical inactivity, and then repetitive head injury from sports. How is AD treated? There is difficulty with treatment due to several factors, including that neurons are formed before and after birth, so there is little or no regeneration throughout a person's life. Therefore, when the neurons are damaged, Unlike other cells in the body, there are no new neurons made to replace them, reducing neurological function over time. And finally, each patient develops plaques and NFTs in different areas of the brain, and the extent of symptoms is determined by this. Depending on the location of where the NFTs and the plaques form in the brain determines the symptoms. This is because each lobe of the brain has different functions and can cause different effects. If forming in the parietal lobe, motor skills such as brushing their teeth can become difficult, whereas the occipital lobe is for spatial memory, which impacts navigation. There are currently no treatments to cure or prevent the progression of AD. Treatments can delay some symptoms from getting worse, for example, behavioural, such as insomnia or aggression, or cognitive decline in patients. There are two main types of cognitive medication. The first is acetylcholinesterase inhibitors. The action of these is to stop the breakdown of acetylcholine, which is a neurotransmitter, by acetylcholinesterase, which is an enzyme. This will allow the nerve signals to keep passing to other neurons to increase neuronal response and activity. The second is an NMDA receptor antagonist. The action of this is to protect the neurons from being damaged from the excessive amounts of glutamate, which is also a neurotransmitter, which is produced in AD. Extensive research into AD is ongoing to find improved treatments. When each is discovered, they will have to go through rigorous testing. Clinical trials. They are clinical experiments or observations which answer questions about new treatments. These treatments can be vaccines, drugs, dietary choices or supplements, or medical devices. All safety and patient data must be maintained to show how the treatment works and its effects. Each study must receive regulatory and ethical approval before commencing and then with any further changes to the study. Why do we need clinical trials? We need them to confirm that the treatment is safe, that the dose poses minimal harm to the patient, has the correct dose with the dose giving the desired result or effect, is successful as a specific treatment for the disease and the benefits outweigh the risks, is able to be taken with other drugs, or to give a detailed list of the prohibited medications that could cause bad reactions, and finally, has minimal impact on the body, especially if there are any long-term impacts to the drugs. After preclinical testing, there are 4 phases that involve human participants. There are 3 before a drug is licensed, and 1 for post-marketing. Each has a different aim and setup. Phase 1. These studies last days to weeks. It is the first in-human study and it involves healthy volunteers. There is only a small group used with less than 50 and is testing any possible harm of the treatment, any side effects, and to determine the correct dosage. Phase 2. These last weeks to months. It involves people who are affected with the disease and it can have up to 500 participants. These test if the treatment is effective. They use a placebo as a dummy treatment to rule out any bias and to determine if there are any further side effects. Phase 3. These last months to years. People are also affected with the disease. However, there can be up to thousands of participants. These trials test if the treatment is effective and how they are over a longer period of time in different countries, and test against existing treatments. Phase 4 trials last years, and it's after the treatment has been licensed to give post-market information. These trials can involve thousands of participants. These test what the drug is like over a longer period of time, how it impacts different groups of people, and what it is like when combined with other treatments. Public's perception of clinical trials is a bit divided. There are 3 main reasons why people don't take part. The first is the potential side effects. However, all new treatments are tested rigorously before use in humans to identify possible adverse reactions. Treatments are always given ascending dose to to give the best therapeutic ability without causing severe side effects. And whilst they can occur, each patient on a trial is monitored closely with regular hospital visits. The next is the risks aren't worth the benefits. However, new treatments are always checked to ensure that the benefits outweigh the risks. Whilst risk will always be there, it will be limited to testing to see if the treatment could help those with the disease, and it could also be the only possible treatment available for the disease. What happens when the drug doesn't work? Ineffectiveness could be due to the patient being on a dummy treatment, such as a placebo. These are randomly assigned to some patients in phase 2 and 3 trials to eliminate bias and to ensure that treatment is working correctly. And finally, if the treatment isn't effective, it could also provide information to how to create better treatment and not be a waste of time or effort. The UK Clinical Trials Gateway is a national database that lists all current trials for each disease, their location, and gives more information about why to join a trial and what happens when you're on one. There are currently 42 trials in recruitment for AD and dementia alone. Why take part in clinical research? There are many benefits to take part, including personal ones, such as access to novel treatments, particularly for a specialised disease, enhanced medical attention, monitoring and equipment to ensure that the treatment is working correctly. Specialised medical teams are involved in research, allowing you to get the best care. And for some diseases, such as AD, enrichment through increased social interaction. There are also community benefits to taking part, such as allowing others with the same disease to potentially have access to a new drug, increasing community spirit to improve perceptions of clinical research, and it may benefit future generations, which could involve family members if the disease is genetic. Many charities and healthcare organisations are available for more information. There is a collaboration between National Institute for Health Research, Alzheimer's Research UK, Alzheimer's Society, and Alzheimer's Scotland to make a register of people interested to take part in clinical trials and assist them with finding the best one for them. In summary, Alzheimer's disease is a neurodegenerative disorder with two hallmarks, NFTs and amyloid plaques. It has risk factors including age, but sadly no cure. Clinical research is ongoing in AD to find better treatments, but there are difficulties with enrolment. Participation in clinical trials is needed to bring new treatments to market, but also has personal benefits too. Thank you for watching.